Okay, thank you, Philippe, for this introduction. I'm going to say also some few words because, as Philippe Musette said, I was one of the PhD students in Philippe Kouriski lab. I'm not coming from the École Polytechnique. I'm one of them. But I wanted to say that being in Philippe Kouriski lab during the PhD training, it was not easy, but it was a lot of fun. I met Jean-Laurent Casanova yesterday evening, and we didn't really share our feeling, our thought about being a PhD student in Philippe Kouriski, but it's kind of very funny that he had the same kind of analysis, that yes, it was fun. It was fun because Philippe made his lab in 5 different groups that were working in different topics, and you could just go there and discuss with the people, share ideas, challenging the models, and then proposing even some new experiment. So I would like to take this opportunity to thank you for letting me join your lab during my PhD, to invite me here. It's a great honor, and as well to embark me in this adventure in Singapore, where it has been also a lot of fun. So I'm going to talk about drug eruptions. So it's something that I started 4 years ago, joining Philippe Musette laboratory. Actually, when I joined his lab, I was supposed to work on the population of B cells that some people call regulatory B cells. I'm still working on it, but because I'm curious and because of my training, I look what the other PhD student was doing, and I thought that drug eruption was interesting, but the model didn't really fit with the data. And this is something that I'm going to discuss with you today. And this is why the title of my talk is Drug Eruption Revisited. So what are drug eruptions? So the definition is that they are immunodermatological disease defined by a skin reaction related to a drug intake. So what does it mean? Basically, it means that when you take some medical drugs, you may end up to have a skin rash. Most of the time, it's very benign because you have the skin rash, you stop to take the drugs, and it goes away. And some people have tried to measure the frequency of this skin rash, and a group in Germany published that around 3% of hospitalized patients could develop these things. And in some cases, one of 10,000 patients, this drug eruptions is becoming serious. So as I said, you have the benign forms where you have rash and with or without blister. You just stop to take the drugs and it goes away, and you get some life-threatening drug eruptions that if you stop to take the drugs, it doesn't go away. So sometimes it's even worse. So you get 3 different types that we call the drug reaction with eosinophilia and systemic symptoms that I'm going to call DRESS, the Stevens-Johnson syndromes, and the toxic epidermal necrolysis, or TEN, or LAERS. So as you can see, the mortality increases with the type of the disease. So they have common feature between this drug eruption. The first one is, of course, the skin rash. You get fever. You get a huge proliferation of lymphocytes. So that means the immune system is stimulated and something is going on. One of the strange features is that you get a delay onset. It takes time before these syndromes start, 2 to 8 weeks. As I said, if you remove the drugs, it doesn't go away. It's sometimes even worse. You get an unexplained cross-reactivity with multiple drugs. And very interestingly, you get even relapse. So the relapse happened because the treatment is corticosteroid. So you immunosuppress the patients, so then it goes better. But if you decrease the dosage too quickly, or if you remove the drugs too quickly, then it gets back. Meaning that you don't really need the drugs anymore, because by then, you may think that the drugs has been totally eliminated from the body to have this kind of syndromes. One more thing is that if you are re-exposed to the same drugs or related drugs, the skin lesion appears faster, which suggests a memory type of response. So There are some particular features. For DRESS, one of the key features is that you get proliferation of eosinophils and as well of monocytes. You get targeting of multiple organs like the liver, the lung, the kidney. So the frequency is quite low. It's a rare disease. And for Stevens-Johnson and the TEN, you get involvement of the mucous membrane as well as the skin, and you get necrosis of the skin with these kind of things that the skin is just peeling off. So this is some of the most common drugs that trigger this kind of disease. You get some antibiotics like penicillin or sulfamethoxazole. You get a drug named allopurinol that you use to treat patients that have excess of uric acid in the blood, and some antiepileptic drugs like carbamazepine. So all of them, they have in common this ring-shaped structure as well as sites which are very reactive. But when you look into the literature, actually you can find any kind of drugs that has been reported to trigger this kind of severe form of drug eruptions. And for example, paracetamol is one of them, which is quite common, but you can trigger some of the disease with these molecules. So what do we know about the pathogenesis? So the pathogenesis is still controversial. We have plenty of evidence that there is T-cell activations. So to explain this T-cell activation, people came with the model where you get a direct T-cell drug interaction explaining the huge immune response. But recently, Philippe Musette and others came with a new hypothesis, which I'm going to talk about it later, which is the anti-herpes virus response. So I don't think now that it's totally exclusive. Maybe it happened in the same times, but we need more evidence of that. So regarding the first model, the direct T-cell drug interaction. So all these molecules per se, they are not immunogenic. They cannot trigger an immune response because they are too small. So they need to be associated with some proteins. So then they will be presented by MHC molecules. And there's three models, the APTEN models, the pro-APTEN models, where actually it's a metabolite of the drugs which bind to the proteins, and the non-APTEN models, which has been described by Pichler, where actually the binding of the drugs is through a non-covalently process. So do we have any evidence of these kind of things that will, of course, trigger, as you can see in this cartoon, T-cell activation? Because obviously, whether the drug is binding directly to the peptide or the MHC, or it's just there inside the pocket of the MHC bulging out the peptide, the MHC structure certainly is different. So it's going to be recognized as a foreign molecules, and you're going to have a major T-cell activation going on. So we do have evidence that indeed there is a CD4 T-cell response. There's a lot of literature about that. I'm just showing you one example where T-cell clones were isolated from a patient with a Stevens-Johnson syndromes. And as you can see here, if you had the drugs, you induce proliferations. And of course, for some clones, there was no proliferation. But as well, in this paper, they modified the drug so there is no anymore this hyperreactive group. So you cannot have a covalent binding of the drugs to the MHC or to the peptide. And still, for some clones, this one and this one, it's in gray here, you induce proliferation. So that's why the group of Pichler came with the model of what we call the PI concept, that actually you can have activation even if there's no covalent binding of the drugs. For CD8, we have also evidence that cytotoxic CD8 T cells can lyse target in the presence of drugs. So there's a dose effect here, as you can see here. And of course, if you increase the effector to target ratio, then the efficiency of the lysis is better. So there is plenty of evidence that indeed the T-cell response against the drugs exists. But what do we know about the T-cell receptor repertoire? The diversity, the theoretical diversity based on the gene arrangement, the mutation, and so on, is 10 to the 18 different T-cell receptors. It has been measured by the group of Philippe Kouriski and others that actually the real TCR diversity is around 25 million different T-cell receptors, and we know that the number of T-cells in humans is around 10 to the 15th. So the question that was very striking is, why finally drug eruption are rare disease? Because you would expect that actually this activation of T-cells will occur more often in any kind of individuals. So, as you know, in the T-cell response, you get the T-cell receptor, but you have also on the other part, you have the MHC molecules. So it's possible that among all the MHC molecules here, 3 types of MHC class 1, different genes for HLA class 2, DA, DQ, DP, and so on, that maybe among all the different alleles, like here, for example, HLA-B, so it's a slide, I took this picture from 2005, but I think now there's more than 600 different alleles just for HLA-B class 1. So it's possible that maybe the fact that the drug eruption or the severe form is a rare disease, is a rare phenomena, it's because the MHC peptide complex is a rare among the individuals. And indeed, when people start to look for association between the disease, in this case, the Stevens-Johnson's triggered by one drugs, they found that a very high direct correlation between the HLA and the genetic background of the population. So, for example, here, if you are Chinese and if you have B1502 in your genetic background, you get 100% chance to develop a Stevens-Johnson syndrome if you take carbamazepine. Okay, so it's a very strong association. So it's been confirmed with Thai population and also with Indians. Very interestingly for the Koreans, same disease, same drugs, but it's not HLA-B1502, but it's HLA-B1501, so close enough. Here, Japanese and Europeans, same disease, same drugs, but different HLA, very striking. And finally, the same Taiwanese group associated the severe form of drug eruptions with new drugs, allopurinol, with B5801. So the conclusion from that would be that, all right, T-cell respond to one particular MHC peptide drug complex, and that's why overall the drugs, the disease is rare. So being in Singapore, 70% of the population is actually coming from a Chinese background. So we were able to test one hypothesis by taking blood from healthy donors, and we screened them for the 2 susceptible HLA, which are B1502 for carbamazepine and B5801 for allopurinols. And actually, I was expecting to have a higher percentage of susceptible donor, but it ends up that I had actually a lot of non-HLA B1502 and B5801 donor. But it was still interesting because I could ask the question if this donor could respond to the drugs or not. And just by doing classical proliferation assay, by using irradiated APC with the T cells in the presence of the drugs, you can see here that actually the T cells in the 5 days culture, they proliferate in the response to the drugs. And if you look at a lot of different donors, so it's, of course, there's a control media. This is the index of proliferation because here the level of proliferation differ quite a lot from one donor to the others. But you can see that in all cases, all the donors that we have tested so far respond at least to one drugs that we were putting inside the culture. So going back to this slide, yes, T cells can respond to drugs. whatever is the system of stimulations. But if we have a so quite common T-cell activation for any kind of donor, does it mean that the severe form is due to some genetic predisposition? Because obviously we need to come up with a new explanation. Before I go into this genetic analysis, I'm going to talk about the anti-herpes virus response. And this herpes virus belongs to a family of viruses that a lot of people are infected, a lot of people are carrier. Most of the time is asymptomatic. The virus, once it infects the host, it stays silent inside the nucleus of the host, like under the form of the episome. There's only one virus where you can detect the integration of the virus, which is it's the case for HHV6. And in some situation, what you observe is that the virus, which is silent inside the cells, gets reactivated, and you have a new cytolytic cycle with a new production of virus. Baptiste Janela, a PhD student in Philippe Muset's lab, has looked at the association between the DRESS syndromes and herpes virus in the literature. And what he found is that there are a lot of case reports where actually the clinician, in the course of the disease, they were looking at if there are any virus inside the bloodstreams, and the answer is yes. So for 60% of this report, you can find HHV6 virus in the bloodstream in DRESS patients, but you can find as well CMV, HHV7, and in some cases EBV. So the question now is, do the drugs can really reactivate the virus production from this latency stage? And the answer is yes. If you take a cell line infected with this, in this case it's HHV6, and you had different doses of the drugs, whatever the drug is, what you can see is that you trigger virus production depending on the dosage and the time of the stimulation. So yes, drugs can trigger herpes virus reactivation, at least in vitro. But now, what about in vivo? And I think it was something that started Philippe Muset and his group, and I think that the study that they published 2 years ago is really a very strong demonstration that indeed in this patient, herpes virus is involved and is playing a role. So what they did is that they took DRESS patients, and they looked first of all if there was any, by serological assay, if there was any sign of previous infection with the virus, herpes virus. And so as you can see, all of them were infected with EBV and as well with HHV6, and for some of them, CMV was also present. But when they looked for virus reactivation in the serum in the PBMC, they found that in 76% of the case, you could show evidence of virus reactivation. Most importantly, what they did is that they used tetramer staining with different peptide from EBV, and they looked among the T cells if there was any CD8 specific T cells against EBV inside these patients. And as you can see, in some of them, 21% of the CD8 are actually EBV specific. So because of this result, they came with a new model. I have to say that these models doesn't rule out the anti-drugs response among the T cells. What I don't know is that what role play these anti-drugs T cells. Do they play a role in the early stage, or do they play a role during the amplification of the EBV response? That's something we need to be addressed. But their model is quite straightforward. The drugs can reactivate the virus, in this case EBV, in some memory B cells. So of course you're going to have new virus productions, and that will infect new cells. And then the T cells will proliferate. You're going to have a major proliferation of T cells with release of cytokine and systemic effect. And because some of them will express homing receptor of the skin, you will have most probably the cutaneous eruption observed. Okay, so there is another thing which is very striking in their paper is like they immortalize B cells from the DRESS patients. And with this cell line, they ask if actually the drugs was able to trigger an increase of virus production. And by doing this, they saw that indeed drugs could trigger an increase of virus production, whatever the drugs use, carbamazepine or SMX. But very strikingly, it was not the case if you were using a BEBV cell line from healthy donors, suggesting that actually the drugs here is triggering something very specific of the donor, again leading us to analysis of the genetic of these patients. So here the question, so why finally these patients are predisposed to that disease? So what we did is that we start a genetic analysis focusing first on copy number variants. So copy number variants are just a segment of DNA that can be amplified or actually decreased inside the genomes. And we perform a comparative genomic hybridizations with the 30 DRESS patients that Philippe Musette and his groups gather. And this group is composed of 25 Caucasian, 3 Africans, and 2 Asians. So a very diverse genetic population. And to our surprise, very strikingly, we find that among 100% of these patients, we find 6 locus that were specifically amplified. And among these 6 locus, they contain 8 genes, and for 3 of them, they are entirely inside the locus amplified. So now what we are trying to address is to confirm this finding, this very striking finding with new patients, of course. And we are trying also to understand what roles these genes are playing in the evolution of the response against the drugs. So to conclude, I've shown you that indeed T cells can respond to drugs. It's clearly evidence that the T cells can respond to drugs. Now, because the T cells, whether you're not going to make any DRESS syndrome, so you are a patient, why they can respond, we think that the explanation is coming probably from a genetic difference. I show you that drugs can induce herpes virus reactivation in vitro, so it's most probably happening as well in vivo, but we need to have more evidence of that. The evidence is indirect because you can find, during the process of the disease, you can find virus in the bloodstream, but there is no really evidence that this reactivation really happened in vivo. Thanks to the work of Philippe Musette, we have clear evidence that in DRESS, at least in DRESS, because it needs to be confirmed for other severe drug eruption like Stevens-Johnson's and the Lyell's. So far, there's only 2 case reports in the literature showing that for Stevens-Johnson or for Lyell's, you find herpes virus during the disease. So it could be different in this case. But here in DRESS, definitively there is a specific CD8 response ongoing against the virus. And thanks to our genetic analysis that we need to pursue, we think now that most probably the DRESS is due to an abnormal strong immune response. And this is why, even if anybody can respond to the drugs, only in few cases the disease is really severe. So to conclude, I would like to acknowledge the people that have worked on this project, which is still ongoing, as I said. So the people in my group, this is a collaboration, of course, with Philippe Musette and Pascal Joly in Rouen. And I would like to thank all the people in SIGN that have been involved at one step in this project, without who we will not have any data. Thank you very much.